just heard from Catherine their extraordinary research in which they use fibroblasts from individuals affected with FH to discover the LDL receptor and identify mutations in the gene that cause FH, earned them the 1985 Nobel Prize in Physiology and Medicine. Their subsequent groundbreaking research identified the molecular pathways responsible for sensing of cellular cholesterol and regulation of cholesterol synthesis and LDL uptake. Their work has had major implications for our understanding of cellular cholesterol metabolism and has contributed directly to the discovery and development of therapies that reduce LDL cholesterol levels. Beyond their research, Drs. Brown and Goldstein are widely admired for their dedication to developing the next generation of scientists. Speaking personally, they've always been willing to share their advice and wisdom with me in a very generous way. Their lab continues to be a vital contributor to cutting-edge biomedical research and cholesterol metabolism, and an inspiration to scientists around the world, well beyond the field of FH. Therefore, I'm delighted to welcome Drs. Brown and Goldstein as our keynote speakers for the 4th Annual FH Global Summit.
Thank you. Thank you, Catherine. Thank you, Dan. I didn't think we were that intimidating. And welcome all of you to Dallas, Texas, where all of this work began and where it's continuing. Joe Goldstein and I decided that because of the mixed nature of this audience, that we would try to sort of give a historical perspective of how we were introduced to familial hypercholesterolemia and how we began to try to figure out the cause of this disease, and then go on to the newer work which deals with treatment. So we've titled this Familial Hypercholesterolemia, from cells to genes to drugs. And from the point of view of full disclosure, Dr. Goldstein and I are both on the board of directors of Regeneron Pharmaceuticals, one of the sponsors of this meeting. So these are the coronary arteries of a 40-year-old man who died of a heart attack. And they've been opened longitudinally here so that you can see the deposits of cholesterol that have initiated the narrowing of this blood vessel and eventually led to the blood clot that chokes off the supply of blood to the heart muscle and causes heart failure and eventually death. Now, when this man was born, the coronary arteries that supply blood to the heart muscle were hollow tubes with a muscular, encircled by a layer of smooth muscle. But over time, that artery has become narrowed so that now the channel has been so narrowed that the lumen of the blood vessel, the channel for the blood vessel, is so tiny that it's ready to be occluded by a blood clot and cause a heart attack. Now, you know, we know that this is a long-term process. Even in people who don't have familial hypercholesterolemia, we know that the initial steps in this process begin in the teenage years. We know that because in the 1950s, when American troops were being killed in Korea, these 19 and 20-year-old boys had autopsies, and even people without a genetic defect, one could see the deposits of cholesterol already beginning in the teenage years. So this is a chronic process that begins, even in people with normal LDL receptor genes, it begins in the teenage years and eventually leads to this terrible outcome. Now, the cause of this disease is a cholesterol-carrying protein that circulates in the plasma. I don't have to introduce all of you to low-density lipoprotein, or LDL. LDL is a globular particle. It's about the size of a small virus, And each LDL particle has 1,500 molecules of cholesterol inside of it. And the cholesterol is attached to a fatty acid, so they're called cholesterol esters. And then this is surrounded by a detergent coat made up of phospholipids and a protein called ApoB. And you can see the formula for cholesterol, and those of you who have some background in high school chemistry will realize that this is a very hydrophobic molecule. It's basically carbon and hydrogen with only one polar group. And so cholesterol is totally insoluble in water. And yet, it is needed by all the cells in the body. Every cell in our body has cholesterol because it's a required part of the plasma membrane that surrounds those cells. So the body has a problem. All the cells in the body require cholesterol, but cholesterol won't dissolve in water like sugar or salt will. And so how do you carry cholesterol around in the body and distribute it to the cells that need it? And that's why the body packages cholesterol in these LDL particles, because this detergent coat on the outside allows the particles to be soluble and to circulate around in the blood. Now the problem with LDL is that it's very sticky, and when it circulates through blood vessels, if there's any damage to the endothelium, the lining of the blood vessel, the LDL penetrates from the circulation, from the plasma, it gets into the wall of the artery and triggers this process of atherosclerosis and eventually the formation of blood clots and death. Now, this is a picture of Joe and me when we met. And I don't show this picture because we were handsome, although we were. But I show it because we were young. And the work that I'm about to tell you about started when we were both in our very early 30s. And both of us had graduated from medical school. I had graduated from the University of Pennsylvania Medical School, Dan Rader's home, which was the nation's oldest medical school. And Dr. Goldstein had graduated from the UT, from what that time was called Southwestern Medical College here in Dallas, Texas, which was then the nation's youngest medical school. And I thought I was pretty sophisticated, having grown up in a big city and going to the oldest medical school founded by Benjamin Franklin. I was accepted as a resident at the Massachusetts General Hospital, which was a very incredibly difficult residency to get, and to be a medicine resident at the Mass General was a great honor. Except when I got there, I realized that they had accepted this other guy from Dallas, Texas. You know, having grown up in New York and Philadelphia, Dallas, Texas was not exactly my favorite place. The year was 1966, and John Kennedy had been assassinated in Dallas, Texas, 3 years previously. And Joe had grown up in this tiny little town of Kingstree, South Carolina. Anybody here ever heard of Kingstree, South Carolina? One person. Wow. Joe, you got to meet this guy. You know, it's a town of about 3,000 people. And Joe went all the way through high school there and then went to Washington and Lee University and somehow ended up at this Podunk Medical School, Southwestern Medical College in Dallas, Texas. And I couldn't understand why he had been admitted to this elite group of interns at the Mass General. But it was very clear within the first 2 or 3 days that he really knew more medicine than anybody else. He certainly knew more than all the interns, most of the residents, and half of the faculty. So he and I became friends. And we were friends because we liked to talk about the basic underlying physiology of disease. Why does this patient have a heart attack? Why does that patient have pneumonia? Why does this patient have arthritis? And it was through these conversations that we developed a deep interest in the underlying biology of disease. And then we were fortunate enough, both of us, to be accepted at the National Institutes of Health for a postdoctoral fellowship. And there the NIH, as some of you may not know, runs a huge research facility just outside of Washington in Bethesda, Maryland. And they have a big hospital that hospitalizes 1,000 patients. And these patients are brought there because they have very unusual diseases, rare diseases that the scientists at the NIH are interested in studying. So we both went to the NIH as clinical fellows and doing research and clinical work. And there we found a little girl who was exactly like this one. Now I have to say, in all honesty, this is not our patient. We weren't smart enough to have a camera. But this little girl looked exactly like our patient. And our patient was 6 years old. She was hospitalized at the NIH because she had a total blood cholesterol level of about 1,000 milligrams per deciliter. And she was hospitalized there because she had already had several heart attacks. The coronary arteries that supply blood to her heart muscle were already blocked at age 6. In fact, she had had her first heart attack when she was 3. And she had such severe angina, chest pain from poor circulation to the heart, that she couldn't walk across the room without having chest pain and having to stop. And it was obviously a terrible tragedy for this little child. And it was worse because she had a brother who was 8 years old, and her brother was also hospitalized at the NIH, and he had exactly the same problem. Now you can see from this picture that this child had what we know now as the homozygous form of familial hypercholesterolemia. And the characteristic hallmark of this homozygous disease are these bumps that develop in the skin. We call them xanthomas. So if you or I fall down and bump our knee, what happens is that the capillaries in the skin are ruptured and some of the blood gets out into the tissue space. And so you get a black and blue mark. Initially it swells, and then scavenger cells from the blood come in, and they clean up all the stuff from the plasma that has leaked out into the skin tissue. And eventually the swelling goes away, the black and blue mark disappears, and we're fine. But this child, the amount of cholesterol in her blood was so great that whenever she would bruise her skin, the blood would get into the tissue. These scavenger cells from the bloodstream would come in, they would ingest all this stuff, but they got so filled up with cholesterol that they couldn't even get out of there. They just stay there, and they produce what's called foam cells in the tissue. And it didn't take much trauma. If you look at her wrists, you'll see that she has these deposits just around her wrists. And the reason was, in this child's culture, they wore copper bracelets to try to produce health. And just from the rubbing of the copper bracelet, there was enough damage to the skin that it allowed the cholesterol to escape from the bloodstream into the skin and produce these bumps. Now, the same process that was going on in her skin is also going on in her coronary arteries. Whenever the lining of the coronary artery becomes damaged from a variety of reasons, these LDL particles penetrate into the wall of the artery, the scavenger cells come in, they ingest this stuff, and then they become stuck there, and that's what initiates the process of atherosclerosis. Now this, as physicians, it was a very, very frustrating and sad experience for us. The first thing we did with these two children was to put them on a zero cholesterol diet. We figured, well, maybe their body is just absorbing too much cholesterol from the diet. So for 6 months, they ate nothing but fruits and vegetables, rice and vegetables. And their cholesterol level didn't drop one iota. It was absolutely, so we knew it had nothing to do with diet. We knew it was genetic because the two children had it, and because their parents, each of their parents had what we now know as the heterozygous form of FH. And so it looked like the parents had a single copy of a mutant gene, and the children inherited a double dose and caused this very severe problem. Now, back in the late 1960s, there was nothing we could do for these children. Coronary bypass operations were not being done. Of course, there was no such thing as angioplasty. The cardiac transplants weren't being done. There were no drugs for cholesterol. So Joe and I decided that we would try to figure out what was the underlying defect. Now, the problem arose because Joe had promised his great teacher here in Dallas that he would come back after his training and start a new program in medical genetics. That's how he got his residency at the MGH, by the way. I didn't have anybody. Nobody was fixing it for me. But he had made this solemn oath that he would come back to Dallas. So if we were going to work on this disease together, I would have to come back to Dallas. And it wasn't too much of a problem for me because I recognized what a fantastic medical school we had here. The problem was my wife. My wife was born in New York, and I'll tell you, when we lived in Bethesda, Maryland, we thought that was the Deep South. And the idea of moving to Dallas, Texas in 1971 was the furthest from either of our minds. But I knew it was a great opportunity for me to work with Joe and try to figure out this disease. And somehow I convinced my wife to move to Dallas, Texas. Of course, I promised her it would be for only 1 year. And that was 45 years ago, and we've raised two beautiful Texas daughters. So, the disease that we wanted to solve, as you very well know now, was not very well known then. In fact, it was called something totally different. It was called type 2 hyperlipidemia. It didn't even have a name. And, but you all know the genetics of this disease. And what attracted us to it as a scientific problem, not only because of our compassion for these children, but because we thought it was a solvable problem because it was caused by a single gene mutation. We knew from the genetics that there was one gene that was causing this whole thing, one mutant gene. You know, genetics, medical research is like looking for a needle in a haystack. But if you work on a single gene disease, then you know that there's a needle in that haystack. And you know there's only one needle in that haystack. And so all you have to do is find the one needle, and you're done. So we decided to try to work on this disease, and as you see from this chart, it compares the heterozygous and homozygous forms of familial hypercholesterolemia. The homozygotes, as you in this room I'm sure all know, are very, very rare. The children that we saw are only 1 in a million children. Their plasma LDL level is at least 6-fold above normal, and they typically have heart attacks around the age of 5. But their parents are heterozygotes, and the heterozygotes are much more frequent. 1 in 500. Now, the new data with all the now that we have the gene and we can make a genetic diagnosis, it looks like it may even be higher than 1 in 500 people. They have the heterozygous form, and their LDL level is not as high as in the homozygotes. It's only about 2.5-fold above normal. But the typical age for myocardial infarction is 35 to 45 years. So what was known when we began these studies? Well, the major thing that was known is shown on this slide. This is the biosynthetic pathway by which the body makes cholesterol. And it had been known for many years that humans and other animals can make all the cholesterol that they need. And they make it from this little 2-carbon building block called acetyl CoA. And what you see here is that it's a very long process. It requires 20 different enzymes to take this little acetyl CoA and polymerize it so that it becomes this 4-ring structure of cholesterol. And that pathway had been worked out over the preceding 20 years by brilliant, really brilliant biochemists. And all of us owe a tremendous debt of gratitude to these biochemists who had figured this out. And it was also known that the rate of this process was controlled by the level of one enzyme, shown in red, the enzyme HMG Co-A reductase. HMG Co-A reductase, as you see, is a very early step in the pathway. In fact, it's the first pathway that's the first intermediate that's committed to cholesterol synthesis. And I mean, it produces the first intermediate, mevalonate, which has no other fate except to go to these isoprenoids that eventually lead to cholesterol. And mevalonate, and what was already known was that this process was subject to feedback regulation. So if you take a rat and you put the rat on a diet with no cholesterol, the rat liver will make all the cholesterol that the rat needs. And it will have very high activity of this enzyme, HMG Co-A reductase. But if you feed a rat cholesterol, the cholesterol will get into the tissues, and the tissues now no longer need to make their own cholesterol, so the HMG Co-A reductase activity goes down. So when we began these studies, it was known that there was a feedback inhibition of HMG Co-A reductase, that the body didn't accumulate too much cholesterol because once it began to build up, it would turn off this enzyme and the body would stop making it. And so that led to our hypothesis. And the hypothesis was very simple. We thought this huge amount of cholesterol in the body must be caused by overproduction of cholesterol owing to a failure of feedback inhibition of HMG Co-A reductase. So that was how we began this work. The hypothesis wasn't really unique because, you know, we knew it had nothing to do with diet. Where was all this cholesterol coming from? You know, that was not particularly special. But was unique or different about what we did was our approach. Because rather than studying the children, who would be extremely difficult to study, we obtained skin biopsies from some of these children, and we grew their cells in tissue culture, in petri dishes. And we compared the cholesterol metabolism in these cells from the FH homozygote patients and cells from normal people. And the gamble was that this feedback inhibition of HMG Co-A reductase would be seen in these isolated skin cells, just as it happens in the whole body of the rat. And in fact, that's what we found. So we took cells from patients with FH and homozygotes and normals. And if you look first at the pink points, the vertical axis shows the HMG Co-A reductase activity in extracts that we made from these cells. And on the horizontal, we're showing the time course. So at 0 time, we harvest normal cells, and you see that they had very low levels of HMG Co-A reductase activity, only 2 units. But if we then remove the cholesterol from the culture medium, now you have to understand that cells are grown in the presence of serum, and serum contains cholesterol. So if we, in these lipoproteins, so if we remove the lipoproteins from the serum so that the cells would be deprived of cholesterol, sure enough, in the normal situation, the amount of HMG Co-A reductase activity rose by 50-fold over the next 24 hours. Well, that was clear then. The cells were being deprived of cholesterol, and they started producing their own. That makes sense from what we knew about the feedback inhibition. And then, as shown on the right panel, we added cholesterol back. After depriving the cells of cholesterol, we added cholesterol back. Well, we had removed the cholesterol by removing the lipoproteins, so we decided to add cholesterol back by simply adding LDL, which was the major source of cholesterol in the serum. And sure enough, when we added LDL back, the HMG Co-A reductase was rapidly turned off. And we thought, well, obviously LDL is somehow donating its cholesterol to the cell. The cell now has an external source of cholesterol. It doesn't need HMG Co-A reductase anymore. But look at the yellow line. Those are cells from a child with homozygous familial hypercholesterolemia. And you can see on the left that at 0 time, she had, even though the lipoproteins and the cholesterol were present in the culture medium, the HMG Co-A reductase in that cells was 100-fold above normal, 100-fold above normal. And we removed the LDL from the medium, there was no increase. And as shown on the right, when we added LDL back, there was no reduction. Well, we were packing our bags for Stockholm. I mean, we thought we had made, this would have been the first example of an allosteric, of a genetic defect due to a loss of feedback inhibition of an enzyme. It would have been incredibly exciting. And we really, really thought we had something terrific and that would be easy to understand. Unfortunately, we did one more experiment. And that is, instead of adding cholesterol back in the form of LDL, we decided, well, why don't we just add cholesterol? Forgetting about this lipoprotein carrier, we'll just add cholesterol back. So as shown on the left, in the normal situation, we had removed cholesterol from the medium, so there was very high level of HMG Co-A reductase activity. And when we added LDL back, it turned off the enzyme. Well, we knew that. And when we added pure cholesterol, it also turned off the enzyme. Great, it's what we expected. But look at what happened on the right side. When we added LDL, the HMG Co-A reductase activity did not go down. That we'd already shown that. The FH homozygote cells don't respond. But when we added pure cholesterol, the FH homozygote enzyme was turned off just as it was in normal cells. So we tore up our tickets to Stockholm, and we realized we had a much, much more difficult problem to solve. Because how in the world does a cell get access to the cholesterol that's inside of the LDL particle? Obviously, the problem, once cholesterol got to the enzyme, it would turn it off. But the problem was somehow the FH cells couldn't get access to the cholesterol that was in the core of the LDL particle. And the conundrum was, well, how does a normal cell, how does a cell get access to this cholesterol? I showed you it's sequestered inside of this globular LDL particle. It can't just jump into the cell. How does it get there? So what we did was to take LDL and to attach a radioactive tag to it. We attached an iodine-125 radioisotope to the protein part, the ApoB of LDL. And then we incubated that with cells. And what you can see here is on the left, when we incubated normal cells with this radioactively tagged LDL, and then we washed the cells and asked how much LDL could bind to the surface of the cell, the normal cells were able to bind the LDL at the surface, and we knew this was specific binding because if we put an excess of unlabeled LDL in the medium, the unlabeled LDL would compete for the radioactive LDL, and therefore the amount of binding of the radioactive LDL was reduced. But as you see on the right, when we added the I-125 labeled LDL to the FH homozygote cells, there was no binding. And therefore there was no competition by the unlabeled LDL. So this presented us with a real challenge because Joe and I had been trained in biochemistry and dealing with enzymes and proteins, but here was a problem that was in cells, and we knew very little about the field of what's called cell biology. But over the next 10 years or so, we were able to work out what was wrong with the FH homozygote cells. And so this slide really summarizes about 10 years of work. And what we found is that when we added LDL to normal cells, the LDL would bind to a protein on the surface of the cells that recognized the LDL particle. And we called that protein the LDL receptor. And we found that the LDL receptors were clustered in a region of the cell, of the membrane of the cell, that was indented and had this coat on it, this fuzzy coat. And those things are called coated pits. And when the LDL bound to the receptor in the coated pits, the LDL was actually taken into the cell, delivered to a digestive organelle inside of the cell called the lysosome, and there the cholesterol was liberated from LDL and could then be used by the cell to make plasma membranes. And we also found something that will be much more important when I finally sit down and Dr. Goldstein gets his chance. He's very impatient, I can see. The LDL, the cholesterol derived from LDL now has regulatory functions. First of all, it turns off HMG Co-A reductase. Well, I told you that's how we got here in the first place. And it also turns down the production of LDL receptors. So there's this feedback mechanism by which cholesterol, the cell maintains an optimum cholesterol level. And when it gets too high, it turns off cholesterol synthesis. If it's too low, it activates cholesterol synthesis through an activation of HMG Co-A reductase. And the problem in the familial hypercholesterolemia cells was that they couldn't produce any LDL receptors. So they couldn't take up LDL from the culture medium. And as a result, they couldn't derive any cholesterol from LDL. So what happens? The cell now has a cholesterol deficiency, and the HMG Co-A reductase activity rises dramatically to compensate for the decrease, and there's increased cholesterol synthesis. So these findings then explained what was going on, what the LDL receptor did. Its job was to reach out into the plasma, pick up LDL, and carry it into the cell. The next challenge was, well, what does this LDL receptor really look like? Because remember, it was sort of a theoretical construct. We had theorized that there was a receptor that did this, but then it was up to us to actually isolate this receptor. And it took another 5 or 6 years to actually isolate the protein. And when we isolated, because plasma membrane proteins are very difficult to isolate, when we isolated the protein, we were then able to isolate the gene. And when we did that, based on the sequence of the gene, we could deduce the structure of the LDL receptor. And so this is work from the mid-1980s, and we showed that the LDL receptor has different parts, and it spans across the plasma membrane. And that green horizontal symbol is it stands for the plasma membrane. And the important thing about this receptor is these repeated sequences that are at the top, numbered 1 through 7, That's the part of the LDL receptor that recognizes the LDL particle, because remember, the LDL receptor doesn't, there are millions of things in plasma, but the LDL receptor only binds LDL. And it's because of this part, this 7 repeated sequences. I'm not going to go through all of the components of this receptor, but it was a big advance when we found it in terms of understanding the structure of membrane proteins. This was one of the first plasma membrane proteins to be isolated and cloned. So now we had the structure of the receptor, we had the gene, and then we started looking at families. And we had developed by this time a huge collection of cell lines from patients with familial hypercholesterolemia, mostly almost all FH heterozygotes, because they had been sent to us by physicians from around the world. And the LDL receptor gene has 18 exons that actually code for these protein regions. And you can see here, I'm not going to go through all the symbols, but there are many different kinds of mutations that can occur in the LDL receptor gene. And in fact, most families are transmitting a different mutation. Unless you come from a place like French Canada, where the whole society seems to be descended from an ancestor that had an FH mutation. Otherwise, the mutations in different patients are totally different. But what they have in common is that they impair the function of the receptor and cause the LDL level to rise. So, you know, I'm talking about studies that began around in the early 1970s, finally culminated with this collection somewhere around 1990. And so at that point, we pretty much understood at least the chemistry of the LDL receptor and and now Professor Goldstein will tell you about the biology and how one can manipulate LDL receptors for therapeutic purposes.
All right, everybody now can tweet about the FH Foundation. And Mike Brown's great talk, although he does exaggerate some of the beginnings. So I've heard it, as I told Dan Rader, probably 101 times. That's okay. So let me begin just to review the hallmarks and the discovery of the genetic basis of FH before going on to how the receptor works in the body. So there are two giants on whose shoulders Mike and I stood before we really got involved as a clinician in Norway named Muller, who is the first one who ever described the syndrome of familial hypercholesterolemia. And in 1938, he described heterozygotes in several families. And he also recognized in these families that the high cholesterol level predisposed the affected individuals to coronary artery disease. That paper is really a classic, but it really, in many ways, because where it was published in a European journal that was not read by people in the US and Britain and France, really did not attract a lot of attention. But the syndrome then received, I think, a great deal of attention with the publication by Awardees Kashadurian from Lebanon of the existence of the FH homozygotes. And Mike described about homozygotes. And Kashadurian observed a large number of homozygotes in Lebanon because there's frequent marriage between two first cousins. So that's called consanguinity in the language of the geneticist. And therefore, that would bring together two heterozygous genes that were identical, and one would then have a large proportion of homozygotes in a population where the heterozygotes were carrying the familial hypercholesterolemia gene. And then as Mike mentioned at the NIH, Mike and I saw 2 patients with FH homozygote, who are FH homozygotes, and that stimulated us to think about how one might approach this disease. And then we went on to show the defective regulation of HMG Co-A reductase in FH cells, and then defective binding of radioactive I-125 labeled LDL in the FH homozygote cells. And then the receptor was purified. This took at least 5 or 6 years. And then once one had a pure protein, one can then use that information to clone the cDNA in the gene for the LDL receptor. That was done in 1984, and that allowed us to identify the first mutations in patients with FH. And we worked on this for about 5 years until about 1990, and after we did about 100 mutations, which were on that slide that Mike showed, we decided that we had done enough. And pretty much there were mutations of every different type that was spread over the whole gene, and we would then go on to other things that I'll tell you about in a moment. I think there are now as many as, there are probably 1,500 different mutations in the LDL receptor gene. Essentially every family has a different mutation, affects a different, alters a different part of the LDL receptor gene. And there are now websites that keep a record of all these different mutations. So now let me tell you how the information from these original tissue culture studies were used to conceptualize and develop a drug for FH. So this slide shows a very simplified illustration of the metabolism of LDL. And for all practical purposes, relative to familial hypercholesterolemia, the liver is the key organ. So the liver makes a lipoprotein particle called VLDL, which contains primarily triglycerides, and it also contains cholesterol bound to fatty acids or cholesterol esters. And that VLDL circulates in the blood, and it's acted on by a series of enzymes in adipose tissue in a process that's called lipolysis. Dan Rader has worked for many years on this and discovered one of the key enzymes involved in this lipolytic mechanism. And the result is that most of the triglycerides of the VLDL particle are removed, and they're then broken down, and they're fatty acids in a adipose tissue where they're then what's called reesterified to new triglycerides, and that's why people get obese if there's too much of this lipolysis and reesterification going on. But once the VLDL is acted on in the adipose tissue and the triglycerides are removed, the remaining particle is called IDL, which is a cholesterol ester-rich lipoprotein that has a very high affinity for the LDL receptor. In fact, it has a higher affinity for the LDL receptor than does LDL itself. And the affinity is so high that in a normal individual, it never circulates in the blood. So therefore, when you have blood tests, you never measure the IDL level. It's just not there. And except for a very small proportion of the IDL that is not taken up by the LDL receptor is converted to LDL. So LDL is basically the remnant of this VLDL process. And then the LDL also binds to LDL receptors, as Mike mentioned, and LDL circulates in the blood until it's removed by receptors, and I'll have more to say about that in a moment. Now in the FH homozygote that has no receptors, I show only for simplicity the homozygote with no receptors, then the amount of VLDL produced by the liver is the same as in a normal individual. The processing in adipose tissue, this lipolysis process, is exactly the same as in a normal individual, and the production of this IDL is the same is in a normal individual, but here's where the difference comes in. Because there are no LDL receptors, the IDL can't be taken up by the liver, and it's converted to LDL. And then there are no LDL receptors in the liver, so the LDL remains in the plasma. And that's basically a simplified scheme of how LDL builds up when there are no receptors in the body. Now, there are several ways to measure LDL receptors in a living individual, and one is that we did the first test assay that we developed after we understood about the fibroblasts was to take blood lymphocytes from individuals and incubate them with radioactive LDL and measure how much LDL they took up and degraded. And you can see in the blue, normal individuals have about a four-fold range of activity. And then if you see in yellow, the FH homozygotes that have no receptors have no detectable activity. And let me just say, give you a little side anecdote in the Mike Brown fashion. We were very lucky in our studies because it turns out the first 4 FH homozygotes we ever studied, remember we didn't know anything about what the defect was, we knew nothing about the different types of mutations. All of ours happened to be FH homozygotes that had no detectable receptors. And I'm not sure exactly whether we would have been able to figure this out had we had what's called compound heterozygotes, where one parent transmits an LDL receptor gene that's not completely non-functional, and another parent transmits another LDL receptor gene, a different mutation that also has a little bit of activity, and these individuals could end up with 20 to 30% of activity that's not too different from a heterozygote with 50% activity. So in the early stages of trying to work out a process, it would have been very hard for us to tell the difference in 20 and 30% versus 50%. So for some reason, we were lucky that we had homozygotes that had no activity. And then it created a problem later on, because then when people tried to reproduce our result, they didn't have homozygotes that had no activity, and they said they couldn't reproduce our result, that they had individuals that had LDL receptor activity. So we had to go through that series. But it was all sorted out when the genetics and the gene was figured out and one understood this variability. Now notice in the middle, the FH heterozygotes has, on average, half the normal number of LDL receptors. And then if you look on the next panel, panel B, there's another assay one can use. Is the most sophisticated of all assays, and Dr. Scott Grundy in the audience here today was a great collaborator of us in doing these studies, where one injects into the bloodstream a radioactive LDL particle, and then you measure its disappearance from the blood over about a 20-day period. And you can see in normal individuals, the LDL disappears, and it's gone by about 16 days, and it has a half-life. Half the particles have left the circulation, remain in the circulation at about 2.5 days. And notice in the FH homozygotes in yellow, at the end of the experiment, there's still many LDL particles in the blood, and the mean lifespan of a particle is 6 days, and the heterozygote is in the middle. Now, if you think about these two experiments, one can conceptualize a potential therapy for FH, because if you look at the heterozygote, who has half the normal number of receptors, that individual has one non-functional LDL receptor gene, but also has a second functional gene. That's why they're heterozygotes. So if one could stimulate the activity of the functional gene by 1.5 to 2-fold, then one could, in theory, create a situation in which the individual would actually express the same number of receptors in the liver as a normal individual, and would therefore remove LDL at the same rate as a normal individual. And so, and this is we came up with this concept as a result of the feedback system that Mike mentioned to you. And as he also mentioned, the cholesterol that comes in from LDL inhibits the HMG Co-A reductase. And so that led to the idea that if one could get an inhibitor of HMG Co-A reductase, then one in this feedback system would actually increase the number of LDL receptors in order to get more cholesterol into the cell to provide the cell with its normal amount of cholesterol so that it could get sufficient cholesterol for its membranes and for its cell division. And while we were working on this feedback system and conceptualizing this idea, again, another incredibly lucky event happened. The appearance on the scene of Akira Endo and his discovery of the first statin at the Sankyo Drug Company in Tokyo. And this statin was a very potent, competitive inhibitor of the enzyme HMG Co-A reductase. So we were very excited by this, and Endo actually had read all of our papers, and he was trying to relate his work to our work, and we knew we could relate our work to his work. So within a year, we collaborated with him and did a key experiment that demonstrated that if you treated the cultured cells with his statin, one could actually not only inhibit cholesterol synthesis, as he had already shown, but one could increase the amount of LDL receptors. So this was sort of the beginning of the idea of statin therapy for heterozygous familial hypercholesterolemia. Again, showing you this right here. So once one came up with these tissue culture findings, then one wanted to see if this concept worked in living animals. And so we and other drug companies tried this drug in small animals like rats and mice, but it turns out rats and mice have no LDL. So essentially you couldn't really determine whether there was an effect of the drug. And so we had to go to actually beagle dogs. And this was I think the first and most dramatic experiment that was done to show that statin administration to an animal would actually would raise the receptors in the liver, shown in the panel on the left, and that increase in receptors in the liver would lead to a removal of LDL from plasma through this radioactive turnover study, like I mentioned earlier, and the removal from plasma was associated with a decline in LDL. And so this study then stimulated us in collaboration with Scott Grundy and David Billheimer to do experiments in FH heterozygotes and FH homozygotes who were treated with maximum doses of lovastatin. And you can see, if you look on your right, the FH homozygotes, again, these were our homozygotes that have virtually no receptor activity. Because they have no receptor activity, when one gave this statin and decreased their cholesterol synthesis, there was no way that, since they had no receptors, that one could compensate by increasing receptor activity, so the LDL level did not fall. And notice in these three panels, the scales are quite different, as you'll see. And then the FH heterozygotes who have one normal gene that could be stimulated, their LDL dropped, as you can see, about 20 or 30%. And then the normal individuals, these would be garden variety individuals who have no abnormal LDL receptors. They have two LDL receptor genes that could be stimulated once one gave statins. Then there was a larger decline when one has maximal statin therapy. So I think this was a key experiment, and then many subsequent experiments were done that confirmed this, and subsequently the statins were approved by the FDA for the treatment of heterozygous FH and severe forms of hypercholesterolemia around 1986 or 87. So if statins can lower LDL, would they prevent heart attacks? And that was the next series of studies that were done in the field. And this is a meta-analysis of 170,000 participants in 26 randomized 5-year trials, and this is assembled by two outstanding epidemiologists in Oxford, Richard Pito and Rory Collins. And the results summarize that they showed a decrease in heart attacks directly is proportional to a decrease in LDL. There's no lower threshold for LDL cholesterol, and I'll show you in a moment what I mean by that. Even if you start out with a low LDL cholesterol, if you are put on a statin in this study, you can still have a decrease in your frequency of coronary disease. All of these randomized trials are done for 5 years, and they're all started generally on middle-aged individuals who are at risk for coronary disease. And then the third conclusion is no significant toxicities or increase in deaths from cancer or other diseases. There are a lot of individual reports of various things, side effects of statins in very small studies without really appropriate controls. But remember, in these meta-analyses, one is talking about, in this particular case, 170,000 participants. And I think it's quite important there was no increased deaths from cancer or any other diseases. So let me just show you, of these 26 trials, there are 5 I'll point out. The first one in yellow is the 4S study that was done in 4 countries in Scandinavia. That was the first study by Merck, and it was one in which the individual started out with the highest level of LDL. The LDL level is on the horizontal axis, and the frequency of major coronary events over the 5-year period from the beginning to the end of the study is shown on the vertical axis. So you can see a rather dramatic drop in the frequency. This is where the concept, the idea of a 2 milligram drop in LDL leads to a 1% decrease in coronary event. And then the HPS study in blue started out with people of about 120 to 130 milligrams per deciliter, not nearly as high as the 4S, and their LDL dropped to around 90, and you can see a major decrease in coronary events. But the one that's quite interesting and really has been important conceptually is this TNT study, the one in purple, where individuals clearly started out with essentially a normal LDL level at 90, and over the 5 years, their LDL dropped to a little bit below 70, and there was also a 3% drop in their coronary events. Remember, these are thousands of people, and all this stuff is highly statistically significant. So the result of all these meta-analyses, especially the TNT and HPS study, has led to the concept about lowering LDL. Over the last 30 years, it used to be a focus on how low, but now it's a focus on how long. So, and the reason for that is because these studies with statins are only for 5 years. In other words, the patients were all middle-aged, and as Mike pointed out, the atherosclerotic process is well advanced by middle-aged and people at risk. And so this brings us, I think I need a new battery. This brings us to the next chapter in the story, PCSK9. So PCSK9, I think everybody in this room is familiar with. The gene was identified and discovered in a French family with familial hypercholesterolemia, and the PCSK9 gene contained a alteration in an amino acid that changed the properties of the normal PCSK9 gene. So the next question was, how does the PCSK9 gene work normally? How does this gene normally affect LDL levels in individuals. When it's mutated, it clearly raised the LDL to look just like a person who had decreased LDL receptors. But how did it normally work? And the original idea from the French and their collaborators in Montreal was that somehow it affected the production of VLDL. But that turned out to be incorrect. And the real, I think, breakthrough in how PCSK9 worked was a definitive study carried out by Jay Horton, who you'll hear tomorrow. And I won't go into the details of his study. I'll let him tell you his story. But he did a parabiotic mouse experiment in which one overexpresses PCSK9 in one mouse, and you connect the circulations to another mouse that doesn't overexpress PCSK9. And if the PCSK9 leaves the liver and goes to the other mouse, then it'll have an effect in the other mouse. It's a very elegant technique that was worked out many years ago, and Horton used this technique. It was the first time the technique was really used in the lipoprotein field, and he was able to conclusively demonstrate that PCSK9 was primarily made in the liver. It was a secreted protein, and that once it got into the blood, it destroys LDL receptors and raises the LDL. In fact, if you remember that diagram that Mike showed you of all the different modules in the LDL receptor, what Jay Horton and subsequently Helen Hobbs and Jonathan Cohen showed that there was one region in the LDL receptor where the PCSK9 bound, one of the middle regions, and it took it to the lysosome, which it normally doesn't go to. The LDL goes to the lysosome, but the receptor does not. The receptor recycles to the cell surface. And Jay will tell you his story tomorrow. But that was a very important study, and at the same time Horton was doing these studies in Dallas, Jonathan Cohen and Helen Hobbs did a now classic genetic study in which they were able to show in the Dallas Heart Study that individuals that had mutations that lowered the activity of PCSK9 led to a prevention of heart attacks. And again, Jonathan Cohen will tell you that whole story tomorrow. I'll show 2 slides from his work because it's so important, but you'll hear the whole story tomorrow. But the combination of the fact that PCSK9 was circulating in the blood and the fact that it destroyed LDL receptors, and the genetic finding that individuals that had low PCSK9 from the time of birth were protected from heart disease stimulated virtually every major drug company to begin a program on PCSK9 inhibitors, and there were antibodies. And as you all know, 2 of them have now been approved, Regeneron and Sanofi, and Amgen, that were approved in October 2015, and there are several others. Pfizer and other companies have antibodies and other approaches to lower PCSK9. So let me just show you one important study from the Dallas, one important piece of data from the Dallas Heart Study from Cohen and Hobbs. Mutations in PCSK9 that lower LDL receptors over a lifetime reduce heart attacks. So Cohen and Hobbs found 2 different mutations in about 3,500 people in the Dallas Heart Study. One was present in 1 out of 30 Caucasians, and it lowered the LDL 15%, and it led to a 45% reduction in heart attacks. These were in middle-aged individuals from 50 to 65 years of age. And then they found in African Americans a more severe mutation in PCSK9 that lowered the LDL by 30%, and that was associated with a 90% reduction in heart attacks. And the way they were able to figure out that these mutations actually affected heart attacks was in a collaboration with a genetic epidemiologist, Eric Bullwinkle in Houston, who was involved in a study in which they followed thousands of middle-aged individuals over a 10 or 15-year period and could quantify the number of coronary events it had during this period of time. And they had DNA samples on all these individuals. So when Hobbs and Coynes identified these two different mutations that lowered PCSK9, they were able to take the DNA from the Bullwinkle collection and sequence it for these two mutations, and that's where these figures came up. And again, this study was, together with the idea that PCSK9 was in the blood, was a tremendous stimulus to the pharmaceutical company. And within 10 years after these studies were done, one had FDA approval of PCSK9. It's the fastest example that I know of translational medicine from the bench to the bedside in this modern era of molecular medicine. One other important thing that came out of the study was that PCSK9 mutations are superior to statins at the same LDL level. So if a PCSK9 mutation lowered the LDL by 15%, then the coronary disease was lowered by 45%. But if a statin lowered the LDL by 15%, remember that's only in a 5-year study, unlike PCSK9, which is one's entire life, then coronary disease was only lowered by 15%. And if the PCSK9 mutation lowered the LDL by 30%, it lowered the heart attack rate by 90%, whereas in the statin it was like 30 and 30. So this, again, is one of the bits of data that suggests that it's not only important to think about how low one gets an LDL, but also how long one needs to treat in order to prevent coronary disease. So in 2015, Regeneron, Sanofi, and Amgen in the New England Journal of Medicine published their papers of their anti-PCSK9 antibodies, and the results were virtually the same. Standard therapy were in hypercholesterolemia patients, individuals treated with statins and/or ezetimibe and diet, and there you can see their LDL levels were 120. And then when anti-PCSK9 therapy was added to standard therapy in both studies, the LDL was subsequently reduced to 50. And both companies did preliminary coronary event studies, and these were not blinded there, and there are a small number of people, about 1,000 people in each group, and not for a long period of time, but they were preliminary, and in both cases, both companies got essentially the same result. There was about a 2-fold difference in the number of coronary events. Both studies now are doing outcome studies that are 5 years in duration, that are looking at thousands of people, and questions may come up about the nature of these studies in the question-answer period, so we can wait for the details about that later. So towards getting ready to the end, towards the end of the last century in 1996, Mike Brown and I were asked to write an editorial in Science on to outline the progress and promise of research in coronary disease in the preceding 20th century. And we entitled our editorial Heart Attacks Gone with the Century and a question mark. And we deliberately chose this title to be provocative. And remember, this was in 1996. Our aim was to challenge scientists to abandon bandwagon research on tired topics in favor of pursuing new areas in a more original, intense, and passionate way. And it's gratifying to us that in the 20 years since this editorial was written, one totally new approach that no one envisioned, anti-PCSK9 therapy, suddenly emerged 10 years ago. And if coronary disease is to be ultimately prevented, we need more out-of-the-blue discoveries of the PCSK9 type. So 100 years from now, when historians write about medicine in the 21st century, hopefully the chapter on atherosclerosis will be entitled Heart Attacks Gone with a Century with No Question Mark. And I would hope that a picture in that chapter would show an FH homozygote, which I think was a big stimulus to the development of both the statin therapy and the PCSK9 therapy. Thank you very much.
So we're going to invite anybody who wants to ask a question. There are microphones at the front of the rooms here. I'd invite you to come up and avail yourself of this fantastic opportunity. But while people are coming to the mics, I want to ask you a first question, thank you, which is why do we have PCSK9? What's the physiologic role of PCSK9? And since it seems like you can upregulate the LDL receptor with statins and other approaches without any consequence, why do we need PCSK9 to regulate the amount of LDL receptor?
It's the answer is totally unknown. You know, we spend a lot of our time trying to psychoanalyze God. I mean, why did she put PCSK9 on this earth? Okay, I had a pet theory, which the experts in this field tell me is all wet. But what PCSK9 does is it very selectively destroys LDL receptors in the liver, which basically means that you're redirecting LDL someplace else besides the liver. And one good place is the ovary. The ovary needs a lot of cholesterol to make estrogens. And also the placenta. When babies are developing, they take up huge amounts of cholesterol from maternal plasma. So I thought that might be one reason that one would make PCSK9, basically to divert cholesterol away from the liver. But Jay Horton tells me that's not true.
So another, so the whole feedback regulatory system, which we did not have time to go into, involves a pathway that Mike and I and our colleagues have worked out called the SREBP pathway, and that pathway turns on the LDL receptor and PCSK9 basically at the same time. So that's another complexity. So you're turning on a protein, PCSK9, that's destroying another protein that you're turning on. So it's conceivable that somehow we're not smart enough to figure this out, that one has to have the perfect balance. And there's a third or a fourth component that's missing in the whole thing that regulates those two also. So there's still a lot of work to be done. What is your idea, Dr. Rita?
I wish I knew. I was hoping you would tell me and give some good ideas.
But it does look like that the LDL receptor really is its major target. It doesn't have what's called major off-target effects.
I guess there are a couple of the receptors that have been shown, at least biochemically, to be targeted in the family, right? But how the physiologic importance of that, I suppose, we don't really know. So I got an email from a colleague this morning. He put a high-risk primary prevention patient who had an LDL of in the 90s on 10 of atorvastatin, and 6 weeks later, or 2 months later, the LDL was 15, which is a little bit of a robust response. Right. But of course, his question, which we've all been asked many, many times, is, can you have an LDL too low? And should we be concerned about people who have very robust responses to LDL reducing therapy, or just say, go for it, that's a great LDL to have?
We have come to accept as normal LDL levels that are really totally abnormal for the human species and certainly for other primates. So when we're all born, our LDL level is about 50 milligrams per deciliter. If you measure it in the umbilical vein, 50 milligrams per deciliter. That's at a time when our body, the fetus has just been developing and needs lots of cholesterol. The LDL level is 50 milligrams per deciliter. And if you stay in a society where you eat very low cholesterol food, such as traditionally in China or rural Japan, the LDL will stay below 100 milligrams for the rest of your life. It's only in our Western societies where the LDL builds up because of the diets we eat, which deliver cholesterol to the liver and suppress LDL receptors. So the question is, what's the normal LDL level? Is it 120 as it is in the US, or is it 50 that when we were born? And we, how many years ago, Joe, when we calculated what the affinity of the LDL, we know the affinity, we know the concentration of LDL at which the receptor operates the most efficiently. Okay, because if LDL is too high, it saturates the receptors. And it turns out that we calculated that we thought the physiologic level of LDL was about 25 milligrams per deciliter. So based on what the LDL receptor is designed to see. And that's about what you see in monkeys. So my view, we don't have enough experience with people with very low LDL levels, and obviously we'll get some experience. But right now, it looks, I'd hesitate if you're going below 20, but around 50 probably certainly has no demonstrated toxicity.
There are a handful of individuals who have two PCSK9 genes that don't function, and they presumably they're adults and they've had LDLs of around 20 to 30 all of their life, and so far there's no problem that's been seen. One of them, I think, is a yoga instructor. One is what is the other one? A ballet dancer.
Question here.
First of all, thank you for your work.
Please, and if you could introduce yourself with the questions, that'd be great.
Phil Mendes from Chapel Hill. We often think about FH classically as kind of three big buckets, where there's very isolated groups with the homozygote, heterozygote, and normals. But it seems to me that what you described earlier, there's really a spectrum of disease. I was just wondering if you would kind of comment on the variation maybe across the population.
Well, there is, if what you are referring to, there are people who have two abnormal LDL receptor genes that can look like a FH heterozygote who has one gene that doesn't function at all. Those are what these are called, we're getting to technicalities now, but these are called compound heterozygotes. So they have two partially damaged disruptive genes, and so therefore they're not really true homozygotes. And the genetic definition of Mendel, a true homozygote, is an individual that has two copies of the same identical genetically altered gene. These individuals would not have that, and they often look like heterozygotes who have a complete deficiency of one gene and then one normal gene. So there is a spectrum in that sense. But in general, though, I think that as Mike showed in those pictures, a homozygote in general can be picked up from the fact that there's LDL levels above 400 from the time of birth, and has these cutaneous xanthomas that are never seen in heterozygotes.
But I think in terms of the spectrum, also there's a spectrum among FH heterozygotes,
Right.
A very broad spectrum of LDL levels, in part because of different mutations may only partially inactivate the LDL receptor gene, but also because other things regulate the amount of LDL receptors, diet for sure, and it's not just a fixed thing. There's a spectrum.
Mike, could I follow up to that? If a patient had an LDL of 200 and a documented LDL receptor mutation that was causal, versus someone who had an LDL 200 and didn't have a mutation, i.e. didn't have at least documented FH, are they different? And would you treat the FH patient more aggressively because of the lifetime exposure?
I think I'd certainly treat both of. I mean, if I were practicing, which I'm not, for purposes of honesty, but it would make sense to treat both of them aggressively if their LDL is 200. But you're right. The prognosis is worse for the person who's had it since childhood, and that's why finding FH youngsters is very important. I think it's still controversial as to what age you would treat an FH heterozygote child. I don't know what your practice is, Dan. I mean, do you have an age cutoff or?
Well, there's several pediatric lipidologists here, but I usually around 8 or so if the LDL is high enough. Yeah.
I mean, the shocking and surprising thing for those of us who grew up holding our breath when the statins were developed is they do seem, other than the muscle problems that we all know about, there doesn't seem to be any long-term toxicity so far. And now we're talking, the first statin was approved in 1987, so we're talking about 30 years of statin therapy. And we haven't seen long-term toxicity. When we were working with Merck to try to develop the first statin, there was a lot of pushback from physicians because, cholesterol synthesis is too important in the body. If you block cholesterol synthesis, you're surely going to have some catastrophic effect. But what I think they didn't understand is that you don't have to block cholesterol synthesis very much. All you need to do is to lower it enough so that the LDL receptor gets upregulated. And so there really isn't a profound decrease in cholesterol production in patients taking statins, and presumably that's why we don't see a lot of toxicity.
Josh?
You may need to hit the button on the top.
Speak loudly.
Just
You could use this one if you like.
by the way, this audio company comes from North Carolina, not Texas.
Josh, come. Okay. Yeah, Borge, why don't you go ahead, and then Josh, we could come to the podium maybe. Okay.
That was an amazing talk, Josh Knoll, Stanford, and the FH Foundation. Last year we heard from Dr. Cacciaduri, and so to see the incredible role, that the personal role that FH patients played in your research was quite amazing and moving. And that was a comment. And then the question is, over the last couple of years, there's been an emerging role of statins in diabetes, the pathogenesis of diabetes. And I'd heard an apocryphal story that at one time you both predicted that might be the case, and that you observed that FH patients very seldom got diabetes. And I'm wondering whether that's really true, and if so, why do you think that is?
That was Joe's observation.
Well, so I would like to hear what the clinicians here think. I mean, when I was seeing a number of FH patients, I really never saw any of them that were actually obese or had diabetes. But remember, this was 35 to 40 years ago before the real increase in obesity. So what do you think, Dan and others? Do you see obesity in FH heterozygotes?
Yes, certainly. We certainly see heterozygotes who are obese and insulin resistant. But I'm rather compelled that statins do seem to modestly increase the risk of progressing to diabetes, and I think we don't really understand the mechanism. I don't know if you believe that or if you have any insights into the mechanism.
Well, if you look at the data, as far as I reviewed it, they don't take somebody with normal glucose tolerance and suddenly make you diabetic.
No. Right.
The studies show that if you're really sort of on the borderline, it can push you up,
Right.
And my view is that if you're on the borderline anyway, you should take a statin. But it doesn't just suddenly make somebody require insulin.
Yeah.
It really is almost, it's like a diuretic. Diuretics will enhance glucose intolerance, yet, if you have congestive heart failure or or hypertension need a diuretic. I
Absolutely.
think you have to take a diuretic.
Clinically, you wouldn't not use a statin because of that.
Right.
Borge?
So now, okay. So, I mean, based on your research, fantastic, and many people that follow you in this room and elsewhere, we know exactly, I think, very well how to diagnose FH. We know what it comes from, and we actually are quite good at treating it today. But despite that, so I'd like to hear from your perspective, why is it then that even in this country, patients are not being diagnosed and they're not being treated? I mean, what are your thoughts about that?
Well, I think one needs a major education program, and hopefully it might, with several major pharmaceutical companies coming out with new treatments, this should be an impetus with the FH Foundation, which was not present way back in the beginning of statin. So I think that combination hopefully will lead to an improvement in the diagnosis. Most people really probably don't know they have FH until somebody in the family has a coronary event. I think that's probably the clearest way, unless the family already has been diagnosed with FH and then other people get tested. But if you don't know about FH in a family, that's the way it's usually picked up rather than by a blood test. And there are discussions about whether everybody should have an LDL level measured early in life.
So if I may follow up on that, I mean, again and again, we hear that there's a misconception amongst non-specialists that FH really is rare and that they're very busy with lots of other pressures, and so they're not looking for a rare condition. And, you know, I think we need to look at the medical textbooks, because it's my understanding, even though I didn't go to medical school, that it primarily shows young children with xanthomas, et cetera, and really the most extremely impacted individuals where it is rare. So, you know, I hope that we can work together to really start to address that significant misconception. I know it's not the only driver, but I don't know if you guys agree, Dan. And
let me say, first of all, I want to congratulate you and the foundation for, because you really are changing the equation. It really is a tremendous thing. You know, very frequently, you know, there are two stages of where diseases are recognized. One, before there's a therapy, you have groups formed, but they're generally like Alzheimer's disease. We have no therapy, but there's an Alzheimer's disease association hoping to find research that'll give some answers. In the case of FH, it's a different story because now we have a treatment. So it really is important for this foundation to do its work. And that's why Joe and I are here, although it took 4 years.
it did take 4 years of me asking. And you know what? We had to bring the barbecue to them.
But anyway, the fact is, to this day, you read articles, the cholesterol hoax, you know,
Yes.
why cholesterol is oversold, and, you know, it's only being promoted by drug companies to make money, okay? Nobody makes money on a statin anymore. But the fact is, you know, it just pains me to watch. These people who write these articles are actually doing a disservice. They're actually causing deaths because people listen to them and then they say, oh, I don't have to worry about my cholesterol. I just read this article in the New York Times. So they're as guilty of causing deaths as anything, and just like the people who are against vaccines. And so we need people to speak up and talk about where the science has gotten us and what science says.
Yep. Christy?
So, Joe, you finished up talking about going forward, more novel therapies, and just get your thoughts on the role of genetics, some of the more recent population studies. GWAS thinks new gene targets. I remember, I think in 1984, I did my senior residence conference here at Southwestern on a patient with very low HDL cholesterol, and you came up afterwards and you said, Christy, HDL is complicated. So we've looked at the genetics of that, and you should study something else. So
Well, I was right.
you were right then, so now
I wish I had talked to them then, Christy.
So I'm going to ask you again now. So we have a lot of exciting things going on. In terms of genetics, once again, ApoLA,
Right.
PTL3, PTL4, lots of targets. Do you want to throw out where we're going to go in some of these areas?
Well, I do think you need to have a clear-cut genetic single gene disease in order to really work out a target and how it works. I think it's very hard in the GWAS because it's the changes are so small. And, as I mentioned before, even in the heterozygous FH, and the system is regulated, if one didn't know anything about it, I'm not sure Mike and I could have figured out FH on the basis of heterozygotes if we didn't have homozygotes.
Right.
Function. You think those are the way?
Right.
Well, they're there, but, boy, you're going to think I have a one-track mind here, but the fact of the matter is that if you look at populations where LDL levels are low, okay, even though they have all these other polymorphisms, let's take the Hobbs-Boerwinkle study of the ARIC study of African Americans who have PCSK9 mutations, and they only, their LDLs are about 100, okay? They're not 50, they're 100. But before the age of 60, they basically have a 90% reduction in heart attacks, 90%, even though they have diabetes, they have cigarette smoking, they have hypertension, they have ApoC3 abnormalities. If the LDL, it just seems to me that the instigator of the disease is LDL. Now, if the LDL is low enough, then no matter what else is happening, you're not going to have atherosclerosis as we know it. But once the LDL reaches above some level, then these other things affect it. So, you can look at, there are people who have LDLs of 200, and I was once in Toronto, in Canada, and they took me to see an 85-year-old woman who had heterozygous FH, who had been documented to have LDL levels over 200 for, as long as they had measured it. And she was in the hospital because she was having cosmetic surgery to remove the tendons and xanthomas on her Achilles tendon so she could get her ballet slippers on at 85 years. So yes, there's no question that there are other factors that influence whether any particular individual is going to have a heart attack. But if the LDL is low enough, then those factors we don't think, I don't think play a role.
Eric, I'm not sure that mic's working, but give it a try.
Does it work? Yeah.
Yes.
Yeah, it does. Thank you very much for a beautiful overview of your work. And closely related question to the last one. In these families with loss of function mutations, do you see low levels of Lp(a), and do you expect that that's a part of the explanation that the effect is so big in these families?
So, well, I don't actually know the answer to that.
Well, it's surprising. I mean, there's something that we don't understand for sure. Statins don't lower Lp(a), but anti-PCSK9 does. And, we could hand wave as to what the explanation is. I don't know. But again, I would say that even if you have Lp(a), if your LDL level is low enough, then the chances of all the epidemiologic data and the genetic data says the chance. Look at people with Tangier disease. Okay, no HDL whatsoever. Okay, you'd think that they should be like FH homozygotes, but they're not. Okay, and the reason people cite is because their LDLs tend to be low. So if your LDL is low, you don't need HDL, and I don't know that you need ApoC3 either.
Yeah.
I'm not a physician, but I'm a patient of Dr. Josh Knowles, and my untreated LDL was 385. I'm 70 years old. My father passed away at 86, and people ask me, how come?
Well, that's a good
You're
question.
absolutely
Okay.
Well,
No, I was taught you're like this lady in Toronto, and the fact is there are people like you, and having an LDL of any certain level is not absolutely predictive of having a heart attack. Okay, it's not, because different people have different thresholds. But the way I view it is that if we don't know. I mean, we don't know. Let's say you were 50 and you had that 385, and yes, your father had very high LDLs and lived to be 90. I'm not sure I'd put my bet on that. I'd probably take a medicine.
I'm not taking any chances.
good.
Kat?
Good afternoon. Thank you. I'm Kat Davis Ahmed from the FH Foundation. I also have FH, and I want to thank you so much for all the work that you've done. You shared with us a little bit about the role, or a lot about the role that people with FH, and particularly homozygous FH, played in your research, and spoke just briefly about the pushback when Merck was developing statins, about the concerns about cholesterol. Were there other roadblocks in the path on the way to statins, and did the FH patient community play some part in that? Can you share a little bit of that story with us?
Sure.
Thank you.
No, there were a lot of roadblocks. I think the biggest one was the Sankyo company, who developed the first statin and began the first trials about the same time that Merck had found their statin and was beginning their trials. The Sankyo company reported that they had seen tumors in dogs that were getting statins, and that required Merck to stop their program. But it turned out, to make a long story short, there's no evidence that there's any tumorigenicity from statins, either in animals or in humans, because you saw the 170,000 meta-analysis. And Dr. Endo, who designed that study in the dogs, it was the first dog study he ever did, he had no idea what dose to use. And what he says, that he was absolutely convinced it was no tumors, it was undigested drug that caused necrosis in the intestine with secondary lymph nodes that got enlarged. And that was part of the reason he actually lost his job at Sankyo, because he challenged them on this. So that was, I think, a big roadblock.
The other roadblock was the view of the physicians. So when Sankyo, when Endo at the Sankyo company found the first statin, they tried to do it because here it is, a discovery in Japan. There was no coronary disease in Japan. So they had to do a deal with a Western company. And the first company they approached was Merck. And Roy Vagelos was, at that time, the head of science at Merck. And Sankyo wanted a trade. They would let Merck develop the statin, but they wanted access to one of Merck's new antibiotics. So Merck sent its marketing team to the American College of Cardiology meeting, and they handed out a questionnaire. As they asked the cardiologists, what level of cholesterol in the blood do you think would justify a drug treatment? And the answer came back 350 milligrams per deciliter. 350. And the marketing people said, go to their actuarial tables, and they say, oh, there are too few people who have that high level. There's no money ever to be made in statins, so our antibiotic is worth much more. And so they didn't do the deal. In fact, Sankyo did the deal with Bristol Myers. And the fact is that, but what Vagelos did do was to keep the research going until they could find their own statin. And of course, it was Merck that introduced the first commercial statin, lovastatin. So anyway, the important so there was a huge prejudice. I mean, England, the UK, one of our AV people is from the UK, a lovely young lady, and the fact is that the UK was the control group. Scott, do you remember Michael Oliver? There was a whole thing in the UK that cholesterol had nothing to do with heart attacks, and they thought it was totally unjustified to give drugs for cholesterol. So you were fighting against this idea that the normal cholesterol goes up to 350.
Another interesting side aspect of the statins was when they, in animals, the HMG Co-A reductase has this diurnal rhythm where its activity goes up during the day, well, it goes up during the night and down during the day. And so for some reason, Merck decided when they did their package insert that once you get the drug at night. And so that led to a series of letters to the editor of the New England Journal that their patients who were taking statins had insomnia. Yeah. And so, fortunately, that was dismissed when a study was done to show it wasn't
Let me
true.
tell you, I was terrified. When that letter came, I said, if there's ever a self-fulfilling prophecy, the drug, here's this medicine. By the way, it may keep you up at night. It's a self-fulfilling prophecy. So,
and it turns out it doesn't matter.
So people stayed up and worried about it.
Exactly. And so, there was concern. Remember in the first label, you had to have a slit lamp examination of your lens of your eye because there was maybe 2 dogs in the Merck toxicity studies that and there was a previous problem with a previous cholesterol-lowering drug that had caused cataracts in the eye. So, when it was first approved, you had to have cataract. I don't know now, and you also had to have liver function tests. Is that out of the label now?
Yeah. Well, nobody has those.
You don't have to have LFTs after 3 months or whatever. So. Anyway, so there was a lot of concern in the beginning. And Merck, and again, I pay tremendous tribute to Merck, even though we're associated with Regeneron. But they were approved by the FDA. Again, just like the anti-PCSK9s, they were approved based on LDL lowering without any evidence of reduction in heart attacks initially. And the reason that happened was because in the 1960s and 70s, there was a trial using cholestyramine that Scott remembers very well. Some of the elderly people in this audience might remember. There aren't that many elderly people here, actually. But that's because it's FH. But sorry.
It's mainly clinicians and scientists. It's mainly clinicians and scientists.
But anyway, but anyway, the fact is that you don't realize Joe and I were always used to be the youngest people in the room, and it hurts us to be the oldest. But anyway, the fact is that now I forgot what I was going to say.
I feel
You're too old.
Cholestyramine. Oh, yeah, because cholestyramine had been shown to lower heart attacks in the rare people who could stay on cholestyramine for several years. And, that was the Lipid Research Clinic's study. And it had demonstrated that lowering LDL lowered heart attacks. And so the FDA approved the first statin based on LDL lowering, but Merck committed to doing a follow-up study, which was the 4S study. And there's something that Joe didn't mention about that 4S study, the first study to show that statins actually reduced heart attacks and actually extended life. What's so impressive about that study, which was done in Scandinavia, if you tried to do that study now in the United States, you'd be put in jail. If you were in Texas, you'd be executed. But the reason that that study was so unethical, they took people who had documented coronary disease, and they had LDL levels of 190. That was the median LDL level in the population, and they put half of them on placebo. Can you imagine taking somebody with coronary disease and an LDL of 190 and putting them on placebo? We can't do that anymore. But back in the when that study was done, which was in the late 80s, early 90s, it was perfectly justified not to treat somebody with an LDL of 190. So it just shows you. And Scott is going to give a talk about guidelines, right, tomorrow. And the fact is that he headed this fantastic treatment panel which recommended the targets for LDL and progressively moved lower and lower over the years until this recent group just blew it all up. But the fact is that the whole society changed in terms of what was considered optimal LDL levels.
Next question.
I'm Susan Campbell with Women Heart, the National Coalition for Women with Heart Disease. And the story you've told took place over several decades with, intermittent successes. And I'm wondering what advice you would give to young researchers kind of to stick with it, and how do you do that?
Joe?
Well, I think the key would be identify the right question and the right problem, and then hopefully one will get a result early on, and then if you get a good result, then stick with it and keep digging deeper and deeper and deeper.
And find somebody to pay for it. I mean,
that's the hard part today.
That is the hard part today. What's really sad about our country is that when we were getting started in the 70s, if you had good training and you had a good question and you wrote your first grant to the National Institutes of Health, you could pretty much guarantee that they'd give you a shot and you'd have a chance. Today, the pay line is so low, the percentage of grants that are actually being funded is so low that young people, what they're doing now is they're staying in their fellowships for longer and longer and longer because they can't go out and get their own grants, and yet the best ideas are when you're young. And so hope something, I hope, by the way, I hope, even though we're simplifying the world by saying if you lower LDL, you won't have heart attacks, I hope that you all realize it doesn't mean that we don't think we need more knowledge about atherosclerosis and heart attacks. And we're not, even though we know that cigarette smoking causes lung cancer, and if you stop smoking, you lower your risk of lung cancer a lot, it doesn't mean that we should stop working on lung cancer. So there's plenty more to learn about this field, and young people need to jump in and do it. And somebody's got to pay for it.
So I mentioned Richard Pito, who is probably arguably the most eminent epidemiologist living today, who has studied both lung cancer and coronary disease. And he actually says that the totality of evidence for LDL as a causative factor for coronary disease is stronger than the evidence that smoking causes lung cancer.
Wow. Yeah.
And that's a pretty, I think, important statement from a prominent scientist.
Okay, we're going to take the remaining 3 questions. So, David.
David Marin at Stanford, thank you for your presentation and your great work. I couldn't resist the opportunity to ask about LDL lowering. Should we aim for a specific target? Should it be different for primary versus secondary prevention? Or should we just be satisfied with a certain percent LDL lowering?
Well, as I said, you're going to hear a lot about that from Scott tomorrow. It all depends on the clinical scenario. In somebody who has other very severe risk factors like diabetes or hypertension, you know.
FH.
Well, FH is a given, but we're I think he's talking about people who don't have FH, who have a certain LDL level. Should we try to get them as low as possible, or should we stop at some level? And it depends on what you think their probability of having a heart attack is.
And also male versus female, male, and also family history. I think those are two things that are frequently overlooked when one focuses on diabetes and smoking. And
so,
well, for a patient with FH,
yeah,
should there be a target?
Oh, sure. I think if you have FH and you're as we said earlier, you've had that LDL level since childhood, and now you're 30, you've already got a lot of damage, and you really need to get as low as you can. That would be my feeling for FH.
Borge?
Yeah, just one more comment. It's about when you asked about these roadblocks on the way on cholesterol. I think one thing that you missed, you should say, is that what we have today, all this talk about statin intolerance. I mean, when I started working in lipid clinics, we had all these questions and so forth. There were so many side effects, and then the statins came, and there was no side effects. Nobody talked about it. And then it came back to us. So I just want to share a little bit anecdote. 2 weeks ago, I went to a meeting in Central Asia, Bishkek. It's Uzbekistan, it's a former Soviet republic. And 2 cardiologists took me around for 2 days, and then there was this young guy, he spoke English, and the older one didn't. So, and nobody in that country speaks English. Nobody is exposed to what we are in the media about statins being bad for you. So the young guy said to me, well, have you heard about this statin intolerance? Because I've never seen any of it. I don't know what it is. And then I said, well, okay, you're a young guy and so forth. And then I asked him to translate to the driver, who had been a cardiologist for 13 years, how many statin intolerance had he ever seen. And then he was sitting there, 5. So he had seen 5 patients. And I asked how many cardiology patients had he been through in his life? Well, about 5,000. So that was 1 in 1,000. So that's maybe the real thing.
I wish that is such a great story. That is an absolutely great story. And, you know, we have to explain why Joe showed this meta-analysis of 170,000 patients treated for 5 years with a statin, and there's no difference in muscle complaints between the placebo when they don't know whether they're taking placebo or not. Now, there are people who will absolutely swear that, and all the clinicians in this audience and some of the patients may have experienced it. Oh, gosh, I took a statin and I couldn't move, and then I stopped the statin and it all went away. But for some reason, we just don't see that. And I can tell you my own personal story. Before I started taking Lipitor, I kept a daily chart of how stiff I felt every morning when I got out of bed, and, you know, after doing my exercise, bike, what I felt like, because I wanted the control in case, make sure that it didn't happen, change. But the fact is, you know, Regeneron did this study, and other people have done it too, right? Where unfortunately, they designed it very, very, very poorly against the advice of some of their board members. But the fact is, they took people who were documented statin intolerant. I think this is published, right? The people who were intolerant to at least 2 different statins. They randomized them to anti-PCSK9 or a statin or ezetimibe. Okay. And they didn't know whether they were getting, you know, it was all well controlled. And about 23% of the patients had their symptoms return when they took the statin, and 23% when they took anti-PCSK9, and 23% when they took ezetimibe. And if you think that the statin and the ezetimibe both were doing this, there's nothing in common. You know, if you say that the muscle thing is due to blocking HMG Co-A reductase, then ezetimibe doesn't do that. If you say that it's due to lowering cholesterol, ezetimibe doesn't do that either. So the fact is, you know, That's the problem is they didn't have a real placebo group. That was the reason the study was. Everybody got something. They should have had somebody on just a placebo, and I think they would have seen the same 23%.
One more question, then we're
Yeah,
going to.
thanks. Edward Yanis, Melbourne. I'm just wondering if you can enlighten me. There's such a vast number of mutations in this LDL receptor genes, and I'm interested in a number of other diseases like, say, hemochromatosis, where there only seem to be a small number of pathogenic mutations. Is there something, is it just that there's so many points where you can make mistakes that are critical, or is there some other explanation?
So my view would be it's not a hot spot in the gene. It's the protein structure of the LDL receptor. It has multiple disulfide bonds with these repeats. And so if you disrupt any of those disulfide bonds or any amino acid between these two cysteines for this disulfide bond, then you affect the function of the protein and it doesn't fold properly. The most common mutations in the LDL receptor are single amino acids that affect the folding properties of the receptor. So I think it's a property, and the same thing with things like von Willebrand's disease, a bleeding disorder there. Everybody has a different mutation, and that gene is constructed similar to the LDL receptor. So I think it's the protein, not the DNA.
Also, there's probably no selection against it because heterozygotes have their problem after their childbearing years. They've already had their children, so there's no selection like in cystic fibrosis or other diseases.
Great. So before we break, Catherine has a few more comments, and we have
and they're not
something
scripted
to do.
No, I would just like to. I know you guys have received the ultimate prize for your scientific work and many other prestigious awards, but you truly are heroes to this community, and the FH Foundation is a partnership. We're a partnership of patients, and I would like to ask all of those here who have FH if you would be willing to stand in honor of the work of Dr. Goldstein and Brown.
Standing.
And yes, and please remain standing. Please remain standing for a minute, please. And I'd also like to ask all of the clinicians in the audience who dedicate so much of their life and career to treating those with familial hypercholesterolemia to also stand. And I would like, please stay standing. Oh, we don't stand enough. That's part of the problem. And I'd also like to ask those who are scientists to please stand to honor Dr. Goldstein and Brown. And everyone else, you really are our heroes. So we would like to, last year we had the honor of giving this award to Dr. Kachadorian. It was our first FH Foundation Pioneer Award, and we would like to humbly give this to you. And thank you.
Very much. Thank you.
Okay. Dr. Goldstein, thank you.